Okay, let's dive into this. So, welcome to this explainer. Today, we're focusing on what is honestly one of the most incredible, high-speed biological processes happening inside your body right this very second, the mechanics of human vision. We're going to essentially simulate this amazing microscopic relay race taking place in the back of your eye. It's this wild process where a single, tiny particle of light gets transformed into the rich, vibrant, moving reality that you're experiencing at this exact moment. Here is our roadmap for today. First, we'll orient ourselves with the architecture of the retina. Then we'll meet the true cellular stars of the show, the rods and cones. After that, we're going to explore this totally mind-bending concept called the dark current paradox. From there, we'll walk step by step through the split-second phototransduction cascade, and finally, we'll see exactly how that tiny cellular signal is sent all the way to your brain. Let's get right to it. All right, section 1, architecture of the retina. So before we even talk about how light is captured, we really need to understand where this is happening. And honestly, the journey light takes is spatially kind of counterintuitive. When you look at the microscopic structure of the retina's 10 distinct layers, light actually passes all the way through the interneural layers, so past the ganglion cells, past the bipolar cells, before it finally reaches the photoreceptor cells located all the way at the outermost edge of the retina. It's almost completely backwards to what you might expect. But light absolutely has to navigate this complex gauntlet to kick off the process of vision. Now, right behind those photoreceptors is layer number 1, the retinal pigment epithelium, or RPE for short. You can think of the RPE as the unsung, hardworking maintenance crew for our vision. It's basically a single layer of hexagonal cells firmly attached to Bruch's membrane, and they do all the heavy lifting to keep things sharp. They renew the photoreceptors, they recycle vitamin A, and they even use melanin granules to absorb scattered light. That last part is super important because it ensures our visual image stays crisp and clear instead of looking all washed out. Moving on to section 2, let's meet the rods and cones. Okay, so the light has finally made it to the back of the eye, and these are the cellular stars just waiting to catch it. And their physical shapes brilliantly illustrate their names. You have the rod cell, which is tall, thin, and very distinctly cylindrical, and then you have the cone cell, which has a much more tapered cone-like shape. Both of these cells have inner segments that house their organelles and outer segments that are just packed with these membranous discs. And it's actually inside those tiny microscopic discs where the real magic of absorbing light takes place. Now, their different forms dictate very distinctly different functions. We have about 120 million rods, and they are incredibly sensitive. I mean, they're capable of absorbing just a single photon of light. This makes them our ultimate dim light night vision specialists. But there's a catch. They only give us blurry black and white images because multiple rods are all funneling their signals into just one neuron. Cones, on the other hand, well, we have way fewer of those, about 7 million, and they require a lot of bright light to work. But because each cone connects to its own single dedicated neuron, they provide the incredibly sharp, high-resolution, full-color vision that we rely on all day long. Which brings us to section 3, and this is wild, the dark current paradox. We've arrived at what is easily one of the most fascinating mechanisms in biology. To understand how we see light, we actually have to understand what our eyes are doing in the dark. We need to talk about the dark current. You see, most neurons in your body rest pretty quietly at a negative potential, just waiting for a stimulus to make them fire, right? Well, photoreceptors do the exact opposite. In total darkness, they are wide awake and constantly active. Sodium and calcium ions are continuously flowing into the cell, keeping it depolarized. Because of this, in pitch blackness, your photoreceptors are endlessly releasing a neurotransmitter called glutamate. So the totally counterintuitive truth of human vision is that light actually turns off the photoreceptor. Think about that. In the dark, ions flow in, and a heavy stream of glutamate is pouring out from the bottom of the cell. But the instant a photon of light hits, those ion channels close, and the glutamate release stops cold. That sudden absence of glutamate, that is the actual signal telling your brain, hey, I see something. All right, section 4, the phototransduction cascade. So, how exactly does light turn this cell off so incredibly fast? Let's zoom all the way down to the molecular level. This is basically a biochemical relay race that happens in a fraction of a millisecond. We can break it down into 4 macro steps. Step 1, the absorption of the photon. Step 2, the activation of intermediate proteins. Step 3, the sudden closure of the ion channels. And Step 4, the hyperpolarization of the cell that stops the glutamate. Let's walk through this sequence. Step 1 happens deep in the outer segment disc membrane. Embedded right in this membrane is a protein complex called rhodopsin. Inside rhodopsin rests a very special pigment molecule called 11-cis-retinol. When a photon of light comes crashing in, it strikes this specific molecule. The energy from that light instantly forces the 11-cis-retinol to physically change its shape, isomerizing into what is known as all-trans-retinol. And boom, that tiny shapeshift sets off an explosive chain reaction. Because the retinol molecule changed shape, the surrounding rhodopsin protein is forced to change its shape as well, transforming into an active state called metarhodopsin 2. Metarhodopsin 2 then basically tags the next runner in our relay race, a messenger G protein called transducin. Transducin drops its bound to GDP, grabs a GTP molecule, and its alpha subunit breaks away, fully activated and ready to hunt. So what is transducin hunting for? Well, it immediately seeks out and activates an enzyme called phosphodiesterase, or PDE6. You can really just think of PD as a highly aggressive cellular cleanup crew. Once activated by transducin, PD starts rapidly hydrolysizing and destroying molecules of cyclic GMP, or cGMP, breaking them down into regular GMP. Now, why does the destruction of cGMP matter so much to us? Well, because in the dark, when cGMP levels are really high, those molecules act almost like physical door wedges, holding the sodium and calcium doors wide open. That's our dark current. But in the light, as PDE destroys the cGMP, the wedges are literally pulled out, the doors slam shut, the influx of positively charged sodium and calcium stops entirely, effectively hyperpolarizing the cell and shutting off the glutamate release. The cell has successfully turned off. Which takes us to Section 5, sending signals to the brain. So, we've successfully converted a photon of light into a lack of a chemical inside a microscopic cell. How on earth does that become vision? Well, through the synapse, this tiny cellular shutoff turns into a macroscopic electrical impulse. Check this out. In millisecond 1, the glutamate level suddenly drops. Now, remember those intermediate bipolar cells waiting right next door? A decrease in glutamate actually removes an inhibitory hold on them. So in millisecond 2, the on bipolar cells depolarize. In millisecond 3, they pass that excitation right onto the ganglion cells, converting the graded signal into a full-blown action potential. By millisecond 4, that electrical impulse is rushing down the nerve fibers through the optic nerve all the way to your visual cortex to be processed. Which really just leaves us with this final thought. Everything we just explored, the intricate anatomy, the backwards dark current, that super fast transducin cascade, it is all happening millions of times a second in your eyes right now, just so you can watch this explainer. The fact that a microscopic drop in a simple chemical like glutamate instantly transforms into the vast, colorful, moving reality you experience, it's nothing short of breathtaking. The next time you open your eyes in a bright room, take a moment to appreciate the billions of tiny cellular relay races that make it all possible. Thanks so much for learning with me today.